Next, let us talk about the complex permanent tissue. Now, we had seen what simple permanent tissue was all about. Now, we have to talk about the complex permanent tissue. We have talked a little bit in the earlier lesson about this complex permanent tissue, and we know by now that there are two types of complex permanent tissue. First one is xylem, that is the water bottle of the plant, and then other one is phloem, that is the tiffin. To be very specific, it carries the water from the roots to the tip, and it carries the food content, whatever is formed in the leaves. Because we are talking about tissue, so we have to deal with the cellular composition as well. We also talked about how complex permanent tissue is different from the simple permanent tissue. I am going to make you recall it, that supposedly this is the part of complex permanent tissue that you are seeing, and this is the part of simple permanent tissue that you are seeing under microscope. Supposedly, the type of cells which are present in the simple permanent tissue is, the cell type is A. So, all the parts will have A types of cells, okay. Now, it depends whether the space is to be kept, how the arrangement would be, but whatever area you are considering, it would have those types of cells only. In the case of complex permanent tissue, supposedly the tissue is made up of three types of cells. One is A, other is B, and the third one is C. So, we have three different types of, just for example consideration, understanding sake I am giving you this way of presentation. A, B and C, three different types of cells are making it. So, the complex permanent tissue arrangement could be like this. I hope I am able to make it clear that all the cells would be present in the given area, as opposite to the case of simple permanent tissue where there was only single type of cell that was present. So, this is the basic difference between simple permanent tissue and complex permanent tissue. Now, talking about the two different types of complex permanent tissues, what we see is that it is having xylem and phloem. Now, xylem is further made up of four types of cells. Now, what are those four types of cells? Firstly, we have xylem vessels, very important for carrying the water or the sap above to the leaves from the roots. Second is tracheids. These are, you can say, accessory water carriers along with vessels, but in certain plants they are the main water carriers. Then we have xylem parenchyma, and then we have xylem fibers. Now, before we go any further, what you have to remember is, except for this part, that is xylem parenchyma, rest all cells that are present in the xylem are dead. Xylem fibers are dead, tracheids are dead, xylem vessels are dead. Please remember that whenever we are going to talk about fibers, they are going to be dead because they will be made up of sclerenchyma. Whenever we are going to talk about parenchyma, as we discussed, parenchyma was the living tissue in the simple case, so this part will be living. These tracheids and vessels, they are living in the beginning, but because their purpose is only conduction, so it is better that they would be dead. They will be forming a lumen, that would be a tube-like structure, and inside that water would be flowing. We need not to have the living part. Okay, in this case we need not to have the living part, and with we, I mean to say we are talking about plants over here, not we humans. The xylem vessels that are required for the conduction, they can also be done if they are good, they can also be good if they are dead, okay. And tracheids, they can also be done because if they are dead, it would be easy. The living part often provides some sort of resistance to the flow of water. You know, flow of water is totally physical activity. Now, it is better if there is no living cell involvement in it, and for that purpose, the xylem is mostly dead except for the parenchyma, which is required for the radial conduction. You will get to know later on again, but you have to remember at this stage that these parts are living while which one is dead. Okay. talking about the xylem vessels, what you have to remember is, now this trachete is somewhat like this pen. Okay, now if I hold it and open it, just take out the refill, you all have pen in your hands, and take out the refill. That empty tube like structure would be the trachete basically. This trachete is going to have small pits. Now how would be the arrangement? Now supposedly this is the trachete that we are talking about, and I am just, because I cannot give a 3-dimensional representation over here, I am giving you the simplest one that I could make. This is what it would be. You can imagine it to be a pen. There would be small pits which would be present. Such would be the arrangement of the pits. Coming to xylem vessels, first of all it is going to have a large lumen as compared to trachete. Secondly, this is going to have lot many pits for connection. So this is what xylem is. Okay. The xylem vessels and trachetes, how they vary from each other. You can put them more concentrated like this and get an idea what a xylem vessel would be. And these are pits through which the water can enter the lumen. This part is the lumen, these are the pits. This is also the lumen. Lumen I could not show over here because it is a 2D place. You can see, take the example of pen. Now take simplest example. In one case, take the simple pen and put holes in a single line, empty its refill, put holes, small holes like this, it would be the trachete. Now take a marker, I would say, a huge marker, empty it and then make so many pores in it, that would be vessel. This is what you have to understand at this level, that how they vary from each other, how vessels are different from trachetes, and fibers, they would be having the same structure, either they could be they would be the fibers, sclerenchymatous fibers that we talk about. So this is what we have to understand about the xylem. Moving further, what we have next is the phloem. Now what is the arrangement of cells in the phloem? Before going to that, we have to see what types of cells comprise phloem. So the first one is, as over here we had xylem vessels that were the main conducting part for the sap or water. In the case of phloem, we have the sieve tubes or the sieve tube cells, the main conduction. Okay. The second one is companion cells. Third one is phloem parenchyma. And the fourth one is phloem fibers. Now, as in this case, we had one part that was living. Over here, we have one that is dead and rest are living. Okay. This one is also living because it is parenchyma, it is bound to be living. Companion cells are living, they are having a particular nucleus. Sieve tubes are living. But these two cells, they are in close proximity to each other, and such is their arrangement that we call that this sieve tube cell and companion cell as sieve tube elements. Okay. Now why do we call them sieve tube elements? What you see in the name of these two cells is that something descriptive is there. First is that in the sieve tube cells, the name suggests it that there would be a tube and it would have sieve in it. Now what is sieve? Sieve is something which is having pores, which enables the movement of something down the gradient, or you can say, take the example of whenever you sieve away the tea. The tea stays in the sieve while rest of the fluid comes in the cup or container where it is being served. So that is the sieve which is present. You must have seen sieve everywhere. You must have performed many activities related to sieve. In the case of plants, that is particularly in phloem, what we see is that there is a tube-like cell, okay, that is like this, tube-like structure, sieve tube cell, which is having pores at its end walls, and that is known as the sieve. So we find the sieve as well as the tube. So it makes the sieve tube cell. Now the last term is that it is a cell. Cell means that it is going to have some sort of cytoplasmic inclusion or cell organelle, to be very specific. So what happens is there is a peripheral cytoplasm which is present. I would make it like this, not over here. There is a peripheral cytoplasm. That means the cytoplasm has been pushed to the periphery and the central part is hollow. The central part is hollow, that is, it forms the tube. There is no nucleus which is present. Now, how does this cell, how can it be called a cell if it is not having a nucleus? It must have some sort of nucleus that should be present, some sort of governing organelle for its working. For that, we have companion cell. Why it has been named companion? Because it is always present along with sieve tube cell. The individual sieve tube cell will have its companion cell which is having a huge nucleus. It is a typical plant cell, nucleus, other organelles, and this nucleus is responsible for maintaining the gradients. So, the companion cell is a true companion to these cells. How? Because it regulates the gradient inside the tube. Now, when the gradient is being regulated by companion cell, it enables the movement of food or rest particles that flow across the phloem. So, that is why these two cells are important, and one is companion and other is sieve tube cell. Okay. Certain books call it sieve tube elements as well because they consider this part as dead, because it is not having nucleus. So, this is one arrangement. Then there would be another sieve tube. This is how the phloem is present, and then it will have its own companion cell as well, and there would be phloem parenchyma that would be present. It could be present like this. This would be the phloem parenchyma. There would be fibers which would be present. So, this is the arrangement of phloem. Phloem fibers are dead. Again, they would be made up of sclerenchyma fibers. Phloem parenchyma would be having parenchymatous cells. These two cells we have talked about. There are specific P bodies, please remember it, P proteins, or you can say P bodies, which are responsible for this particular conduction and gradient maintenance. This is all about the complex permanent tissue. I hope you get to understand that what are the components. Firstly, you have to remember the names. That is very important. Secondly, you have to understand how it is different from simple permanent tissue. Thirdly, you have to understand what is the basic structure, just the basic structure in terms of function. Whatever function is performed by the specific cell inside the tissue, you have to keep in mind that how that function could be performed because of the structural specialization. Because when we talk about permanent tissue, we talk about differentiation. When we talk about differentiation, we talk about some sort of structural specialization that has come, and that has come in the following ways that we have discussed. And lastly, you have to remember what is their function, where they are going to be present.